Hello, welcome everyone to the Janelia and EMBL Bioimaging Seminar Series. It's my pleasure to introduce today's speaker, Carsen Stringer. Carsen is a group leader here at the Janelia Research Campus. She did her postdoc work with Marius Pachitario and Carl Svoboda at Janelia, and before that she did her PhD work with Kenneth Harris and Matteo Candini at the University College London. Here at Janelia, her group develops tools for understanding high-dimensional visual computations and neuronal representation of behavior. Today, Carsen will talk about Cellpose and Suite2p, anatomical and functional algorithms for cell segmentation. Welcome, Carsen. It's great to have you with us, and please feel free to share your slides and take it away.
Wonderful, thanks. And thanks for mentioning, so feel free to ask questions in the chat or unmute yourself at any time. It's more fun if this is interactive and you can get questions that you have about the tools directly to me as one of the tool creators. Okay, cool. So I'm going to tell you today about two algorithms that we've developed for cellular segmentation, Cellpose and Suite2p. So why don't I tell you what exactly is cellular segmentation? So we have this problem, we have this image, and we're basically going to try to find the outlines of each of these cells in this image. And we're going to assign a label to basically every pixel within a single cell gets its own label. And so that's kind of the problem we're trying to solve with each of these pipelines. And so we're solving this problem because many analyses downstream after you've collected neurons or other types of cells require segmentation. So for instance, if you want to count cells, you want to monitor cell shape changes, you want to quantify gene expression, or if you want to record neural activity, you need precise segmentation of these cellular boundaries. And so some examples of there's lots of different cell shapes that need segmenting. So here's an example of some cells. Another example of cells with a different kind of staining that look quite different. Here's some data from Janelia, from Philip Keller's lab, showing these nuclei kind of migrating around. And then also we have, this is an example of neurons that have been stained here and are being used for in situ sequencing at Janelia. And so we have lots of different cells with different cell shapes. And additionally, and so basically we designed Cellpose in order to try to segment all these different cells with a single algorithm so that people don't have to retrain algorithms over and over again on their different data sets. And then additionally, anatomical algorithms are really helpful, but in the case where you also have information about the cells firing. So in this case, for example, I've circled this cell here and you can see these pixels across time have some sort of time trace. There's additional information here in the time dimension that we can also use to do segmentation. So that can actually help us find even more cells than we can find with an anatomical algorithm. So Suite2p also takes into account this temporal information to do segmentation. And so I'll tell you first about how Cellpose works, and then I'll tell you about how Suite2p works in terms of the segmentation. And so to solve this general purpose segmentation across many different cell types, we chose deep neural networks because they can indeed perform segmentation. So people have been using deep neural networks, for instance, like MassCar CNN, to do these segmentation problems, which are very relevant, for instance, for self-driving cars. You need to know which ones are cars, which ones are people, and estimate where they're going in order to not crash the car. And basically, there's this big data set that many of these algorithms train on, which is called COCO. And so this has lots of images with lots of different types of objects segmented in this way. And so we thought, okay, if there if there was a COCO for cells, then we could train all of our a big network on these cells and get an algorithm that's accurate. But this wasn't the case. There wasn't a COCO in a sense for cells. So that's one problem that we have to solve. And then the other problem that we try to solve with Cellpose is we try to, instead of having this architecture which tries to find ROIs and then create masks in this kind of two-step process, we try to create a network loss function, which was more adequately suited for cell segmentation. And so, so we tried to create a better loss function. So I'll go through both of these here. So basically we tried to acquire enough training data, and then we built a deep network with what we thought was an appropriate loss function for cellular segmentation. So this idea that you need lots of training data to get a general algorithm was really inspired by Ann Carpenter's lab, where she collected lots of different nuclear images. And they found that the best model was trained on all of these images and could generalize. So you didn't need to train a separate network on each of these different image types. And so we hoped that this would also be the case for cells, not just nuclei. And so we collected and labeled many cells. And so Mihaly did all of this labeling, all of the circling of all of these different cells. And he circled over 70,000 cells. And we found these images basically by Google searches and some databases, and collated them in order to have a really diverse sampling of different cell shapes. Okay, and then the question is, how do you make a neural network that has an appropriate loss function for cellular segmentation? So you have this image and you want to assign a pixel. You basically want a pixel to say, this is cell 1, this is cell 2, this is cell 3. But you don't want to train it to learn 0, 1, 2, 3, 4, because the position, the numbers of the cells don't matter, basically. Like
Carsen?
Cell 1 is the same as cell 2. Yeah, sorry.
So, sorry, this labeled or this data that you created, is that somewhere publicly available?
Yeah, so in the paper, you can click on, there's a link there to, it's actually cellpose.org/dataset. So all the data is available there. You have to put in your institutional email because we have a license, because we don't actually own the images. So we needed to put them behind kind of a little bit of a, there's a disclaimer there that you basically have to sign and that sort of thing. But yes, they're all publicly available online.
And did you train only on sort of your data or also on Ann's data, on both, or how was that?
We have a nuclear model which we trained on this data and some other nuclear data sets as well that weren't in this challenge. And okay. So that's our nuclear data set. But for the cellular data set, we did not train on the nuclear images.
Okay, thanks.
But you could try combining these two data sets and training them together. If you think there's different features that you think would be helpful to learn for your cells. Thanks, Ulrike, for putting the link to the paper. Okay, so now the question is, so you kind of want to create a map sort of for the neural network to learn. It can't really learn these 1, 2, 3, 4 labels because those are permutable. Like cell 1 could be cell 2. That's not a good loss function for a neural network. So basically, we created these flow fields for each of our cells. So basically, if you have a cell here, you have flows which tell you for each pixel in the cell, what is the direction from where that pixel is to the center of the cell. And so you can see each of these cells kind of has a sink in the center of it, where all of these pixels are going to end up if you pretended like this cell was like a dynamical system and this is its phase portrait. If you run the dynamics, which is actually what we do at the end, then the pixels will all end up in the center of the cell. And so what we found was this kind of flow can work for complex cell shapes that aren't just convex. So it can take into account these kinds of concavities and cells with these little processes. Which some types of shape descriptors have trouble taking into account. So basically, we took our neural network and we trained it. So for this image, we convert all of these cells into these little flow fields, and so we train the network to predict this image, basically, which is it's two things. It's the X flows and the Y flows, but I've combined them here into this flow legend, which is often used in optical flow. So that each color represents a different direction. We also train it. So there's a third map. We also train it to predict the cell probability, which is 0 outside the cell and 1 inside the cell. And so that's what's conventionally used often in cellular segmentation training. Okay, and so once we get the network predicts this flow, and it predicts the cell probability. We basically mask the flows by the cell probabilities, and we run the dynamics based on these flows. So every pixel starts to move according to these dynamics. We run this dynamical system over maybe like 100 iterations, and all of the pixels within a single cell are converged to a single point. And so that's what you can see here. And then we can basically assign all the pixels that converge to a similar position are assigned to the same cell. And so this allows us to kind of take it to it allows us to learn these kinds of more complex cell shapes that would be difficult to segment with other algorithms. All right, so the first thing we tested was whether Cellpose will work with at least with specialized images. So we took this database of 100 fluorescent images of neurons, and we trained Cellpose just on this database to see how well it could segment. And so what we found was that Cellpose outperformed. So the ground truth masks are in yellow and the predictions are in red. And if you look at where the arrows are, you can see some of these errors here. So for instance, Stardust didn't predict this cell, whereas Cellpose predicted it. Stardust, for instance, cut off these dendrites because it's learning these convex shape descriptors, so it has trouble with these kinds of processes. And then MaskR CNN also makes errors, for instance, with merges and some oversplitting here and around here. And so we can quantify the amount of errors that an algorithm makes. So the common way that people do this is they'll basically take the true positives divided by the true positives plus the false positives plus the false negatives and call this their precision. But how do you define a true positive? That depends on how accurate you care about being. So you take this, there's this IOU, which is the area of overlap divided by the area of the union. And so you can imagine if you have two cells which are overlapping with 50% of their area, they're going to have an IOU of around 0.5. And if that IOU is kind of, if you consider an overlap of 50% as good, then you can say that's a true positive if my IOU is greater than 50%. And so basically you can define true positives based on cells that overlap and are detected. False positives are cells that the algorithm detects that aren't there. And then the false negatives are the cells that the algorithm misses. And so you can compute this at various IOU levels, depending on how accurate you are going to, how accurate you want to be when you're defining a true positive. And so we create these curves as a function of IOU threshold. And so you can start at 0.5, and you can see, so green is Cellpose, red is Stardust, and purple is MaskR CNN, that Cellpose outperforms each of these algorithms across IOU thresholds. So kind of, it has more true positives at this kind of coarse grain level. And then even at higher IOU thresholds, Cellpose continues to outperform because it's able to find these fine boundaries of the cells and these processes that these other algorithms might not be able to pick up because of this flow representation. So there's two things. So we think the flows help to get these complex processes, and also the flows help to avoid merges. So I'll show this again, I think, because this is useful to look at. So you see the network is forced to learn very different flows at the boundaries between two cells. And so we think this helps the network to avoid merges more than other algorithms, like, for instance, a conventional UNet, because it's forced to learn these kinds of very different flows at boundaries between cells. Then we tried to scale things up a bit. So we wanted to see, okay, we can do well on the specialized data set. Can we generalize across images with very different properties? And so we took we had 100 images from the cell image library. We had other cells, which we called fluorescent, non-fluorescent cell membrane. We had other microscopy images. We weren't really sure what they were that we found on Google. And then we have non-microscopy images, which are like images of sea urchins and rocks and things like that. And so we labeled all of these cells and we trained the network on all of these different cells. And not cells, all of these different objects, basically. And we found that on this generalist model, basically on generalized data testing, we specifically tested on all these cells, although Cellpose outperformed MASCAR CNN and Stardust on these other cells too, but I'm just zooming in on the cell aspect. Basically, Cellpose outperformed Stardust and MASCAR CNN on this generalized data set. And then if we go back, so we want to know what happens if we go back to our specialized images. Did we lose performance by making our network more general? It doesn't seem to be the case. So I can overlay the plot from the previous slide on top of this plot, and you can see that our performance on this subset of data was just as good as it was if we had only trained on that data, which suggests that the network kind of has a capacity to learn many of these different filters and many of these different, what I would say, like textures inside the cell that it uses to define cell boundaries and differences between cells. So I think that's just, I mean, these other networks can also do it. So I think it's a function, as long as your network has kind of enough filters and layers, in theory, it should be able to start to learn many of these different cell types. So there's no reason, in principle, to train your network on a very specific subset of data if you have lots of different data that you can train it on, and maybe you can make a single model that can work well.
Carsen, I have a quick question.
Yeah.
In the plots you showed here, when two cells overlapped, one of the cells was on top, and the flow diagram for one of the cells was on top, and the other one was cut in two. But in truth, you probably probably have two complete cells, like you have layers and two complete flow diagrams when you're actually training. Yeah.
No, we don't. But no, that's a really good point. So we don't have we don't allow overlaps. And so like Stardust, we basically have to assign every pixel to one cell only. So we don't have multiple maps. So that is one advantage that MASCAR CNN has, for instance. It proposes these regions, and these regions can be overlapping, and then it goes back in and defines the mask within that region. So Cellpose and Stardust don't have the ability to find, to learn these overlaps. But yeah, I think in theory, it would require more maps of at different levels, for instance.
Yeah. So then the piece that's on the other side of an overlap is assigned to a separate cell?
In this case, we try to get rid of those as much as possible, but the ground truth for this data set, we actually curated because they had done that sort of thing where they had allowed them to be separate. But in principle, yeah, we didn't want to, we can't learn kind of across that boundary.
Sorry, you throw it away. Okay, yeah.
Yeah, I think in some cases we kept some of those, and then it's errors that our algorithm makes and also the other algorithms make.
Sure.
Other than
in most cases, it's probably tiny, the piece that's thrown away, so it's fine. Thanks.
Yeah, but it is something to be mindful of if you have a problem with lots of overlaps. If you don't have 3D information, then you probably want to try an algorithm like MASCAR CNN that can take into account overlaps. Yeah, so that's a good question. Yeah, and I think too, we're kind of it's a question in terms of this overlapping problem. Like we're kind of data limited too. So yeah, I think first pass, I think you can do a good job with an automated algorithm like this that doesn't take into account overlaps, and then you might want to manually curate some of the overlaps if you need to.
Yeah, it's actually impossible to always get overlap right. Like there usually isn't enough information in the image.
Yeah.
So, yeah. I think this is amazing. Yeah.
Thanks. Yeah. No. I think without 3D information, it can be really difficult. Okay, so we're showing here now that Cellpose basically here again, so ground truth is in yellow and Cellpose is in red. And so Cellpose is able to segment lots of these different types of images. Some of them are even shells and garlic. So it's able to kind of find these principles of sort of what I would say textures on cells and know what boundaries look like. And another thing I didn't point out, I should have said, I'm sorry, is that when we're training this cell model, some images, we have a nuclear channel. So you see this blue with this green, and we put that into the model. For many cells, we don't have a nuclear channel, but for the ones that we do, we put in that nuclear channel, and that can help in ambiguous cases. Knowing the nuclear information can improve the segmentation. So it's been trained for cells where we do have a separate nuclear channel with that channel input.
But you don't do anything special there, or is it just a two-color image, or?
Yeah, you're absolutely right. We don't do anything special, and we can't, for these images, we don't have the nuclear segmentation, so we can't actually train it to simultaneously do the segmentation for both. We're just using it to facilitate the segmentation of the cytoplasm.
Okay.
All right, so then the question is, so where can we go from here? So one extension we created of Cellpose was to do 3D segmentation. And so and often you have a lot of, for instance, and I mean, it's not trivial, but segmenting 2D data is much faster than segmenting 3D data. You can imagine if a cell is 30 pixels across, then you have to segment it, for instance, on 30 planes in Z, not just a single plane in 2D. And so we wanted to see how well we could kind of generalize our 2D model that's been trained on lots of 2D data to 3D without doing this expensive data collection of 3D data. And so how this works is we take our cells are collected, our cells are collected in a volume, and we have the conventional slices in Z of these cells. And so we'll run Cellpose on each of those slices in Z. But we'll also run it on slices in XZ and also in XY. And so you can see we're showing this example on a relatively isotropic volume where the sampling in Z is the same as in X and Y. And so this is a particular case where I think this type of general 3D segmentation works well. Okay, so we segment each of these planes. And then so we run the flow algorithm on each of these planes. And then we're going to combine these flows. So these flows tell us, these slices tell us the flows in X, Y. These slices tell us the flows in X and Z. These slices tell us the flows in Z and Y. And so this is where we're going to average information. So having 3D information helps with segmentation generally, because you have more information on which cells are on top and below each other. And now we can actually learn cells that are kind of surrounding each other in a sense, because we have this 3D information. Like what we were talking about before, they would be split in 2D, but in 3D, we can actually find these more complex processes. And so basically we average this information. So the X from XY is averaged with the X from XZ, and each of these are averaged to give us a flow. So here's the flow after averaging these flows in X and Y. This is the same plot. And then this is the flow in Z after averaging the Z's from these XZ slices and these YZ slices. And so this is just a single map. I'm showing like blue is your above the cell, and you're going to go the center of the cell is below you, and red is the center of the cell is above you. And so we have flows in 3D now, in X, Y, and Z, and we can run our dynamics in 3D as well. And this gives us an output that looks like this. And I made it rotate in image J. I think what's more revealing is basically in the Cellpose GUI or in other GUIs too, you can have the segmentation, you can scroll through in Z and look at how it looks, and you can see cells that are segmented. And in fact, kind of these cells can be wrapped around each other, and it's able to find these different cell shapes. But I do want to point this out, because this is like commonly asked question on GitHub and in general, is if your volume is not too isotropic, say for instance you have a lot, don't have very fine sampling in Z, it might not be so helpful to use the information to do the segmentation. In fact, it might work better to run the segmentation in XY, get the masks there, and there's a stitching option in Suite2p, in Cellpose, where you stitch masks across planes. And so if you don't have a very isotropic volume, generally that will work better, because there might be too much noise introduced by these XZ and these YZ slices being very different looking and not kind of segmentable by Cellpose. Okay, and so we kind of we benchmarked how well we did here. We labeled the ground truth for this volume, and we found that Cellpose 3D, which is this green line here, outperformed these other algorithms. And so I'm showing here this stitching idea that I was telling you about, where you stitch masks across planes. And so the stitched version of Cellpose does fairly well too, but it doesn't do as well as using the 3D information. And then I also, oh yeah, sorry, go ahead.
Sorry, so with this stitching, because I think they also, yeah, I've seen this now popping up in other places, but I actually don't get it. So if two cells in 3D are sort of very close or almost touching, I mean, it would think it's one cell, or how is there anything clever that it doesn't join stuff that's touching along the z-axis?
Yeah, so that's a good point. It's going to choose the one that matches the highest. So for instance, if there's like a little piece of the cell and it was a little piece before, the highest IOU will be keeping that small piece rather than merging yourself into the big piece. So we use IOU to do our stitching. So that allows small things will stay with small things and big things will stay with big things. If there's a big change in the cell shape over a single Z plane, then yeah, you're going to run into problems. But if the shape kind of changes somewhat smoothly, then you won't have huge problems.
I mean, let's imagine it's a sphere. I mean, then I don't get, I mean, then it's kind of thick in the middle and then thin. So how does, and then maybe there's another sphere on top.
Yeah, so if it kind of tapers, that's why I'm saying, like, if there's enough sampling that you're getting kind of smaller and smaller, then the IOU changes. The IOU will still be largest for that cell with itself relative to other cells. Mm-hmm. But then if you have two spheres on top of each other and they're touching perfectly, yes, then you could have problems. But generally speaking, that's not, at least in these mm-hmm, in this tissue with these neurons, we don't see those.
I see
those problems as much.
Okay, cool. Thanks.
Yeah, it's a good point. I mean, it's not perfect. I mean, using 3D information is better than not using it. But if you can't, then I think it's a good workaround. And indeed, so for instance, for MaskR, CNN, and Stardust, or even like if you have your favorite 2D algorithm, you can do the stitching there. And that's what we did in these dashed lines. But we found that the Cellpose stitched work better than these other algorithms. And so these, to note, the MaskR, CNN, and the Stardust we used are the ones we trained on the same exact data as Cellpose. So Cellpose does a better job of segmenting these neurons than these other algorithms. And then we also had UNets. I trained UNets with the same data set as well as Cellpose, and found as well that they were not as good. So those are in the blue down here. Okay. All right, so the last thing I just am going to show, you can use the GUI inside that we built for Cellpose to segment your images if you want. And one nice thing here is, so you can run the segmentation in the GUI. Right now, I was actually estimating the cell diameter. I didn't go into that, but this is a good point for users. If you know your cell diameter, you should definitely put it into the algorithm, because it can try to estimate it, but it won't be perfect. And so if you know your cell diameter, it's good to tell the algorithm, because that will help it do better. But yeah, then the other thing I'm showing here just now is that you can label inside the GUI. So this is the GUI actually where Mihaly labeled all of these cells. So if you label your cells here, you can send these segmentations to our server, and we can use them to update our model. And so what we've done, so we've gotten around, I went through and manually curated them to make sure that they were, we need all of the cells to be segmented basically, and that wasn't the case for all the images. But we had around 200 new images that people submitted to us. And so I trained a new model that's called Cyto2 that you can use in Cellpose now, that has these new images, that has a more of a range along SNR, for instance. So there's some images with more noise kind of in this data set now. We found on our original test images, it doesn't actually do any better. It maybe does a little bit better at low IOUs, but it may have more cells like the cells that you have. So it's worth trying out if you want to try it out. Okay, and then, yeah, so that's pretty much it for Cellpose. As we created an algorithm that has out-of-the-box performance for many types of images. You can also go to cellpose.org and see if it works on your image. It runs the network in the browser for you, and you can see if it works well before you do a local install. And we also have this way, if you use our GUI, you can send us labeled images that we can then add to our training set. And we also have integration with other platforms. So ImagePy, ImJoy, Napari, and Cell Profiler can all run Cellpose for you. Cool. All right. So that's it for the Cellpose section. Are there any questions on that?
I would still have one. So
that's fine.
Why do you actually, I'm thinking now, have the nucleus and the cell model separate? Could you just train one thing?
Yeah, I don't think we'd lose performance on the nuclei if we did. So one thing with the nucleus model is the images weren't as high resolution. So it's actually trained at a smaller diameter. So the cell images are trained with a diameter of 30 pixels. So this is something I didn't talk about. What we found was that the algorithm works better if it doesn't also have to generalize across size. So we standardize our images to all have the same average cell diameter within them in pixels. So for the cell model, it's 30 pixels, and for the nuclear model, it's 17 pixels. But in theory, we could have trained the model on all the data together, and I don't think we would have lost performance. I think that the network has the capacity to learn that.
Okay. So you don't rescale your images before feeding them into the model? We
do. So sorry, we rescale them so that they're 30. The cells are 30 pixels across. Mm-hmm. And then we also, so that's why if you, as a user, you know the diameter, then we'll rescale automatically using that diameter, the image size.
Yeah, no, makes sense.
Yeah, so we weren't able to get it didn't work as well if it had to also learn sizes of objects. But there is a way to automatically estimate size, which kind of takes the inside of the bottom of the unit to use to help predict size. That can also work. But if you know the size, then it's even easier just to put the size in.
Okay, thanks.
There's a question from Mark.
Hi. So can you hear me?
Yeah.
Yeah. So one naive question would be, could you, in principle, also extend this to use on like cryo electron tomography, like at least for something simple like a vesicle that also has a closed topology? Or what would you say would be like the main limitations to like go to some completely like different, in a way, data type in a way?
Yeah, I don't think I know cryo EM very well, so I'm not sure. EM in general, people have used Cellpose on EM in general for nuclear segmentation, and it's worked fairly well there. So there is this, the flows won't follow things forever. So I don't think it's going to generalize to, for instance, like tracking axons over long distances. So I think that I don't think that will work so well, but if it's a complex shape that's somewhat isolated, then I think it can work relatively well. But again, you would need some new training data probably for what you're describing if it's quite different.
Cool, thanks.
Cool. Are there any other questions, or should I move on to Suite2p?
I think we are good to move on to the next session.
Cool. All right. So I'm not going to go through all of Suite2p. I'm going to go through the cell detection in particular and kind of this question. So we could use Cellpose on 2-photon calcium imaging data. But what we find is that there's many. So here's an example of a zoom in of this calcium imaging data where we kind of selected cell centers based on by eye, basically. This was before we had Cellpose. And what we found was that several of these cells selected anatomically, if you look at their activity, which are these colored traces, they don't seem to have strong activity that's above the baseline activity. So these gray traces are the activity of the surrounding pixels. And so you detect detecting cells, you might be detecting many cells anatomically that aren't necessarily active, and you might be missing cells that are active but don't show up on your mean image, for instance. So this is the mean over all of time. And so basically using temporal information, like for instance, this is the correlation between pixels across time, that can be more helpful to find cells that are active and have significant, basically these significant transients, which are signatures of GCamp activity. And so we kind of summarize this here, kind of the skewness of this trace is how many GCamp activities it has, and kind of finding cells functionally gives us traces with higher skewness than clicking on them kind of structurally or choosing random ROIs. And so I'm going to go into just the cell detection in Suite2p, but just to note, Suite2p takes lots of different data. It has another step, which is important, which I'm not going to go into, which is the motion correction. So when we're doing this 2-photon calcium imaging, we often do it in mice. People do it in other animals too. These animals are awake and free to move as much as they can while they're head fixed. So there's lots of movements of the brain here. And you can see these movements here that you need to correct, because if you don't correct these movements, then you might think that there's activity in a cell when really it's just a cell moving in and out of your predefined ROI. But I'm going to go into the ROI detection, how that works. And then you can also use Cellpose inside Suite2p if you want to now. And then you can extract the signals. Basically, the sum of the pixels that you define to be a cell across time will give you these traces. And then you can output these traces in various formats. Oh yeah, there's a question, Eduardo.
Hey, hi Carsen. Sorry to interrupt you. Could you please elaborate more? How do you perform the motion correction? I think I missed it. Thanks.
Oh yeah, no, I wasn't. Oh, oops, I'm going forward. I wasn't going to go into it at all, but I can go into it a little bit at the end more. But so basically, there's 2 steps to the motion correction. The first step is rigid registration, which is we take the phase correlation between a reference image we find and every frame of the movie. And so the phase correlation is fast to compute in the FFT domain. So that's where we compute it. And so that gives us a rigid offset of the frame. And then there's a second step where we do non-rigid registration, where we divide the frame into little blocks and we compute the phase correlation of each block with the block of the reference image. And so that we have like 64 blocks, and then we upsample those shifts to a full 512 by 512 pixel number of shifts, and then we do a bilinear interpolation to create our new cell, our new image of the cells shifted by those non-rigid offsets.
Okay, so the overall assumption is that the motion is uniform within one frame, correct?
It can move non-rigidly in small sections, but we generally, if it like completely rotates out of plane or moves in Z, those are things we can't take into account. Or if it like completely rotates as well, those are things that would be difficult to fix with this patch-wise non-rigid registration. But a lot of the movements we see that are non-rigid, we can fix with this. Like if the mouse is like moving its head a bit, you'll see some parts of the plane move a little bit differently, and those we can take into account with this.
Thank you.
Yeah, it's a good question. Yeah, so I'm not going to go into this here, but something to be really careful of is especially the Z movement is very worrying. So if your plane is moving in Z, then you're losing your cells in a sense, and you can't correct for it with this sort of thing. So we have ways of estimating motion in Suite2p to try to figure out if you have Z motion. And so I recommend using that or taking Z stacks at the beginning of your recordings to make sure that you're taking into account the Z movement. Because once your cells move out of plane, they're gone. So it's definitely a real problem that people have when they're imaging. Okay, so basically Suite2p does these different steps for you, and there's lots of options in the pipeline for how these different steps are done. And then there's a GUI at the end, you can process inside the GUI too, to basically to view your results across cells, and you can look at correlations of cells across your field of view, for instance, and things like that. Okay, and basically we've tried to make Suite2p as memory efficient and as runtime efficient as possible, so that basically we take these really large recordings and we want to process them in a couple of hours so that we can come back the next day and make hypotheses about what we saw in our data and change our experiments accordingly. And so we think being in the loop and doing fast processing rather than doing your analysis all at the end of all your sessions can help you kind of to do better experiments. And so that's kind of what we've optimized for, is that ideally you can run Suite2p on a conventional desktop computer in a matter of, I mean, if it's a short recording, in a matter of minutes, basically. And we also have now in Suite2p, we have the ability, you can run it on a server. You can send each plane, if you're doing multiplane imaging, to a different server job. And so that makes it really fast. Basically, we process one of our 50,000 neuron recordings. We send each plane to a separate job, process it in like 15, 20 minutes, which is pretty nice. Okay, so basically, if we could do anatomical segmentation. And if you're going to do this, I recommend definitely using the max projection image. So this is the maximum of each pixel's intensity across time. So that's what this image looks like. And from this, you can run something like Cellpose. So this will work a little bit better than using the mean image, because you're getting the max, there could be cells that are active that you'll see better here than you would see in the mean image. And so if we run Cellpose on an image like this, we get 679 cells, which is decent, but you'll see what we get of around double the cells if we run a functional algorithm. And so there are cases where I think Cellpose is good, particularly for imaging experiments. So for instance, in the hippocampus, I think it could be useful in certain cases to run Cellpose. So here's an example of the plane of a hippocampal recording where I've run Cellpose, actually in this case on the mean image. And we do have hippocampal data in our training set for Cellpose. So I think that's part of the reason why it works well. And then also for really deep imaging. So here's an example of imaging deep in auditory cortex. These cells are not necessarily all super active. This is the max projection image, so you can see them somewhat, but it can help to do a good job of segmenting these cells that are kind of have low SNR, that are deep in the brain, and maybe it will make more clear ROIs than if you try to use the correlation approach. We're using correlations of pixels across time, which might be noisy. Okay, and that's just kind of the GUI in Suite2p. You can run Cellpose with this anatomical only flag. Okay, so now how does functional detection work? So basically we're going to take our movie, which is X, Y, T. So we have these time frames now, and this could be quite big. This could be hundreds of gigs. And so the first thing we do is we bin in time to try to make this more manageable in size. And also because we think there's some redundant information. So GCamp, for instance, might decay on the order of 2 seconds, a time scale of 2 seconds. So the activity within 2 seconds will be relatively correlated. And so for instance, if you're imaging at 30 Hertz and the decay time of your GCamp is 1 to 2 seconds, then a bin size, we bin at a bin size of 60, basically. And so this is what this bin movie looks like. You can still see definitely activity of these neurons in this bin case, but we've made this movie more manageable to work with. And then the next step, what we do is we high pass filter in space and time. And so now you can see these cells kind of jumping out at each other. So the high pass filtering in time helps us to isolate these kinds of spikes in activity relative to kind of slower varying signals, which maybe are not these GCam transients. And then the high pass filtering in space also gets rid of kind of slow signals. So generally speaking, we're not just imaging the neurons. There's also like axons and dendrites above and below these neurons that we're imaging. And so these contribute to slow signals that we see in the plane. And so high pass filtering in space can get rid of some of those signals too. So now you can see these cells popping out here. Okay, and then we're going to uniform filter in space, which is equivalent to basically we're just binning and downsampling our image. And so we create these basically these maps at smaller downsampling sizes. So why do we do this downsampling? So what we expect is that a cell consists of pixels that are correlated with each other. And so if I take a square and all of those pixels are correlated and I sum them, then across time, that activity will be high in that square if I just sum everything in that square and collapse it. But if those pixels are uncorrelated to each other, they're just kind of noisy, and I sum them, they're going to basically be 0. And so as I bin and bin at higher and higher resolutions, or at more and more, you'll basically have peaks on these maps according that are cells basically, and then things that are noise are just going to go to 0 because they don't have correlated activity in time. And so we're trying to, yeah, sorry,
but if your bin doesn't completely overlap with a cell, like it has half a cell in it, then you're losing signal, right?
Yeah, so it could be you could have a cell that's kind of in between two bins, but if it's but in theory there still should be a peak there. If there's some amount of the cell there should be enough of a peak. So that is a good point. Like there still will be some activity there. It won't be 0 like noise, but there's actually a parameter here in the algorithm which tells you how big does that peak need to be for me to go in and find a cell. Like basically how much variance does that cell have that in order for me to say I'm going to go take that as a cell. And so that's this threshold scaling parameter in Suite2p. So you can lower that if, for instance, you're not finding cells, and it will go in and find those cells that maybe are small or kind of not quite accommodated there. So if they're big and they're kind of over a couple bins, then you'll eventually agglomerate them at a map down here. But if they're small and kind of divided across things, then maybe you do need to decrease your threshold for finding cells. Yeah, and so this agglomerating basically means like you're summing more and more. So if a cell is bigger, it's going to have a higher variance across time at these smaller maps, because it's going to get bigger and bigger as we sum more and more. And so we go in and we find peaks across these maps at different sizes. And basically we initialize a cell based on the size of the map it's found on. So if it's found on one of these things that has been downsampled a lot, we initialize the cell as bigger than if it's found on a map that's kind of maybe, for instance, just a binning of 6 pixels, where it would be a smaller cell. And so we go in and we find these cells, and then what do we do with these peaks? And so this is basically where okay, so this is kind of an average picture across all of these maps. We don't actually use this in the model, but this is something that shows up in the GUI. So you can see if, for instance, if an area has a cell, for instance, that if there's something that looks like a cell here and Suite2p didn't find it, then it's a good sign that you need to decrease your threshold scaling, because then Cellpose will run longer and find those cells, and or increase the number of iterations Suite2p runs for. Okay, and so this is kind of the last step here, which is we initialize a cell, and then how do we grow it? And so we're taking this, we're assuming that we're finding these sparse signals kind of in this volume. We think they're sparse in space, and they're also sparse in time, these components. And so we're assuming the sparsity in time. So what we actually do, we take this square that I've initialized based on the size of those maps that I was talking about, and we focus on explaining basically only the pixels that are above a certain threshold or a certain percentile. Those are the activity frames that we focus on. So for instance, these red dots here are times when this pixel basically was active. And so we're going to focus on those pixels when we're growing the cell. And the reason we do this is because you can have 2 cells that are kind of overlapping with each other, and they could have slightly different activity. And we want to try to we don't want to focus on the whole trace, which might consist of activity from this other cell. We want to really focus on these transients that we think belong to this specific cell, which are large. So this allows us, basically, I'll show you. So basically we take these pixels, these time points, and we take the average activity of the pixels in the square here and in surrounding pixels, and we see how big they are. And if they're above or below a certain threshold, we include those pixels in the cell. And in this case, actually, the pixels were inside this square that were above this threshold. So basically just this cell was taken, these pixels in here, rather than more of these pixels. And so this kind of assuming the sparsity in the trace then means, for instance, I can come back with a peak that's nearby and I could extract another cell there that explains other activity that could be overlapping with this cell. So that's why we make this sparse assumption. And then for this cell, this is actually a cell that's pretty active throughout the recording. You can see these cells kind of grow. This cell starts to grow actually as we look at, as we average these frames. You can see these little dendrites grow. And so on each of these iterations, we check the pixels within the box and then every surrounding pixel of each pixel to see if they should be included or not. And in this case, there's some pixels outside that should be included. In this other case, there's other pixels as well that outside that should be included. So this allows us to learn complex shapes from these seeds, basically. And so this is now what the functional detection looks like for the same plane I was showing before. So this finds twice as many, this finds around 1,400 ROIs compared to the 700 you find with Cellpose. So we find more ROIs than we did with the anatomical detection. Okay, I think we're pretty much done. I'm gonna stop now. I'll just show, so you can do cell detection. For instance, we get lots of thousands of ROIs when we do these large, these multi-plane recordings. This growing step allows you to find ROIs that have unique shapes. So here, example, are Purkinje cell dendrites and cerebellum. You can look up this paper if you're interested in how they did the imaging and stuff, but basically they use Suite2p to find these dendrites. And then we have some other new steps we've included, but I'm going to skip over this. But I'll show this basically with some PCA denoising, which is now default in Suite2p, you can find more dendritic processes in these cells. But yeah, let me go to the end. We have a GUI basically where you can look at cells like I showed before. We rely on, again, I didn't say this before, we rely on many open source packages to make these pipelines, which has been they've been really amazing to use, and this software wouldn't be possible otherwise. And then, so in conclusion, I didn't show this, but registration is very accurate. I showed you how cell detection works. And so in some cases, Cellpose can be helpful for you. Also, this PCA denoising I didn't go through can help. Yeah, so I skipped over some things there, sorry. But basically, the next thing we're going to add in Suite2p, things we're thinking about kind of demixing overlapping cell signals. Right now, we basically throw out pixels that are overlapping to really make sure that we're isolating cell signals from each other and we're not mixing things. But we've also, we're kind of beta testing this demixing cells. And then also, what's next in Suite2p is potentially something you add as the community. And so for both Cellpose and Suite2p, we take lots of pull requests, and we're happy to add features that people are interested in that can help the community in general. And so I'd like to thank, so for Suite2p and for Cellpose, Marius and I collaborated on this software. And then we also had some help from Nick Del Grosso and Chris Key last summer, refactoring the code, which should make it easier for people to use. And thanks to Janelia for funding all of this research.
Thank you very much, Carsen. That was an amazing presentation. I think this tool is incredibly powerful and also an amazing enrichment for the community in general. And what I like a lot is also not only how it performs, but also that it's really accessible to everyone. That's an open source tool that it can be used either installed on your own computer, or as you've mentioned, that it can be driven also in the browser. So it serves lots of maybe, even if one works in a lab where, I don't know, one's restricted in resources, it can be used. And that's actually quite great. So with that being said, I know that we are kind of running out of time. So I would say we, but also something else, I would still ask people to ask questions, let's say 1 or 2 questions before we actually start with the Meet the Speaker session. So the Meet the Speaker session, I haven't introduced this at the beginning, but Carsen will actually stay on the Zoom call for an additional half an hour to ask more in-depth questions. For example, how exactly you can use this tool on your own data and so on and so forth. So this won't be part of the recording, but please feel free after, as I said, 1 or 2 questions right now, we will dive into that segment immediately. So, but as I said, with that being said, are there right now more questions? Please feel free to raise your hand. Otherwise, I hand over the word to Christian Tischer, who will briefly introduce which talk we can look forward to in December. Christian, please.
Yeah, thanks, Ulrike. Yeah, I think there's one more talk for 2021.
Yeah, that's correct.
Okay, so this will be Nicola Gritti from EMBL Barcelona, and it will be for a change microscopy talk again, because I think we had now quite a lot of image analysis. And this will be about the mesoscopic imaging facility there. So they are imaging stuff that's a bit bigger than cells. So yeah, I think that will be fun. And then, yeah, see you there, and then maybe see some of you in the Meet the Speakers session in a minute or so.
Okay. Thank you again, Carsen. Bye-bye.